Hello everyone. Hope you all are doing good and are safe at home. Today's topic is clinical trial phases. Let's see with introduction. What do you mean by clinical trial? Clinical trial is a systemic investigation in human subjects by evaluating safety and efficacy of any new drug. Clinical trials are any investigation in the subjects intended to discover or verify the clinical, pharmacological, and pharmacodynamic effects of investigational product. Pharmacology that deals with the study of a drug. Pharmacology is derived from the Greek word pharmakon, that is, drug to make or synthesis of drug. Pharmacodynamics is a term used to describe what the body does to drug, that is, the process of absorption, distribution, metabolism, excretion of drug when proceed in our body. Pharmacodynamics is also known as pharmacokinetics, and the abbreviation used for pharmacokinetics is PK. Why do we conduct clinical trials? What are the goals or objectives behind clinical trials? Clinical trials are conducted only when satisfactory information has been gathered on the quality of the non-clinical safety. Authority or ethics committee approval is granted in the country where approval of the drug is sought. That is, the goal or objective behind conducting clinical trial is safety, efficacy, and faith of consumer on medicine. This is the diagrammatic representation of the drug development process. That is the process of synthesis of drug to approval by FDA authority for launching a drug in market. Now let's study all four phases of clinical trial in detail. Phase 0. It is also known as microdosing. It is the new idea from the US Food and Drug Administration. Study of new drug in microdose to derive PK information in human before undertaking phase 1 studies is called phase 0. The objective behind phase 0 is to obtain pharmacokinetic data. The toxicity of a drug has been studied by two routes of administration. It can be intravenous, subcutaneous, intramuscular, or oral route of administration. It is the very early step of clinical trial to obtain pharmacodynamic and pharmacokinetic property of a drug in humans. It helps to choose between competing chemical analogs for further study. Now, phase 1. The aim of a phase 1 trial is to determine the maximum tolerated dose, that is MTD, of the new treatment. 20 to 100 healthy volunteers are participated in phase 1. Do we know why healthy volunteers are participated in phase 1? Let's discuss. Researchers need to compare healthy volunteers with people who have a specific disease or condition. They are tested on healthy volunteers to check that they are safe. The substances are then tested on people with the illness to ensure that they are safe and that they work. Phase 1 is proceed in 3 steps. SAD, single ascending dose studies, MAD, multiple ascending dose studies, food effect. It is the investigation difference in absorption caused by food. Studies are conducted to know the potential impact of food intake on the absorption of the drug. One of the examples for explaining food effect is tetracycline. An antibiotic tetracycline is prescribed to a pregnant woman. It will cause an fluorescent yellow discoloration to babies whose teeth are not fully developed. It has been observed in children below 8 years. Phase 1 is divided into two, phase 1A, phase 1B, where phase 1A is single ascending dose. Now let's get differentiated between phase 1A and phase 1B. Phase 1A, that is small group of subjects given single dose of a drug and observed for a period of time. Each cohort group has given prescribed milligram dose to obtain the MTD. It is the dose which produces therapeutic effect. Cohort can be defined as the same kind of population with same indication. The amount of drug increases with each subject to obtain MTD. Whereas in multiple ascending dose, phase 1B, a group of subjects receives multiple low doses of the drug and the samples of blood has been collected at various times and analyzed for better processing of pharmacokinetic and pharmacodynamic and safety of the drug. As you can see in this slide, phase 2. It is also known as therapeutic exploratory trial. Number of subjects used is 20 to 300. The objective behind phase 2 is to confirm effectiveness, evaluate safety data, to monitor side effects, optimum dose finding, dose efficacy relationship, therapeutic dose regimen, duration of therapy, frequency of administration, therapeutic window. Phase 2 is also divided into two phases, phase 2A and phase 2B, where phase 2A is designed to assess dosing requirement. It means how much drug should be given. Whereas in phase 2B, it is designed to study efficacy of a drug. How well the drug works to a body at the prescribed dose. Phase 2A. Let's get differentiated between phase 2A and phase 2B. Phase 2A. It is early phase, pilot clinical trials. Pilot initiation of the clinical trial. 20 to 200 patients, not multicentric. Single blind comparison with a standard drug. Late, whereas in phase 2B, late phase. Pivotal clinical trials. Pivotal means fixed dose required to produce therapeutic effect. 50 to 300 patients or number of subjects. Multicentric. Double blind compared with a placebo or standard drug. Placebo is a dummy pill or a drug with no active pharmaceutical ingredient API. Phase 3. It is also known as therapeutic confirmatory trial. The objective behind phase 3 is to collect safety data to assess overall and relative therapeutic value of the new drug efficacy, safety and special properties. It is carried in large number of patients. Phase 3 is also divided into two phases, phase 3A and phase 3B. Let's get differentiated between phase 3A and phase 3B. Phase 3A. It is carried out in large number with a special category. It regulatory requirement for NDA, where phase 3B is also known as label expansion. It is performed after applying for approval but before launching. This may supplement or complete the earlier trials or may be directed to phase 4. The objective behind phase 3A and phase 3B is to generate or collect safety data and drug efficacy and special properties of a drug. Phase 4. It is the last phase of clinical trial. Phase 4 is also known as post-marketing surveillance. No fixed duration or patient population. Objective behind phase 4 is confirm the efficacy and safety profile in large population during practice to detect the unknown adverse drug reaction. Adverse drug reaction have a causal relationship with treatment. Evaluation of overdosage, identification of new indication. If any adverse drug reaction has been observed, you can report adverse drug reaction to by a formal reporting system. In India, it is National Pharmacovigilance Program, that is Central Drug Standard Control Organization. The criteria for reporting ADR, that is adverse drug reaction, is identifiable patient, identifiable reporting source, a suspect medicinal product, an event identified as serious and unexpected by drug. Now get revised by all four phases of clinical trial with the help of this tabular chart, that is preclinical testing. Duration of months or years, 6.5 years. Test population is laboratory or animal studies. The purpose behind preclinical testing is safety, whereas in phase 1, duration is 1.5 years, number of subjects 20 to 100. Purpose determine safety and dosage. Phase 2, duration 2 years, number of subjects 100 to 500. Purpose evaluate effectiveness, look for side effects. Phase 3, duration 3.5, number of subjects 1000 to 5000. purpose to confirm effectiveness, monitor site reaction, and long term use. Phase 4, it is also known as post marketing surveillance, additional post marketing testing required by the FDA. Success rate 5,000 compounds get evaluated in preclinical testing. By 5,000 compounds, only 5 compounds can be entered in phase 1, phase 2, and phase 3. And from all above 5 compounds, only one compound is get approved by FDA authority. FDA is Food and Drug Administration. NDA, that is the new drug application, it is the regulatory authority which provides permission to launch new drug in market. An investigational new drug application, that is INDA, it is the regular authority who provides permission to conduct clinical trials. This is the summary of all 4 phases and how the product get launched in market from the synthesis by scientist or researcher. These are the few references which I have referred for this topic. Thank you for watching this video. Hope you like it.